This presentation is a recap of our quiz, the first on-site quiz for Basic Veterinary Pharmacology for first semester 2022-2023. This was administered last September 22, 2022, and the topic coverage is on pharmacodynamics. Students are directed to answer the following questions accordingly. For true or false, write the word true for correct statements and false if otherwise. For question number 1, not all drugs exert their pharmacologic actions via receptor-mediated mechanisms. True or false? So the answer for this question is true, because some of the drugs utilize other means whereby they will be able to exert their mechanism of action. So here we have, for example, cellular mechanisms of action and the non-cellular mechanisms of action. So there are those drugs which acts extracellularly. This means that it will not involve any type of receptor. We also have those drugs that will be able to act to its target tissue or target molecule other than the receptor-mediated mechanisms. For question number 2, the concept of blank explains a maximum response being achieved when only a fraction of the total number of the receptors is occupied. So this concept is also known as despair receptor concept. So this means that for a drug, so not all drugs require that all of the receptors will be occupied in order to produce a maximum response. So some of the drugs in veterinary pharmacology or in therapeutics would only require a fraction of that particular available receptors to produce a pharmacologic response. For question number 3, most receptors are nucleic acids. True or false? So this is considered to be false because most of the receptors are made up of proteins. Question number 4, blank is the concentration, EC50, or the median effective concentration or median effective dose, ED50, of a drug required to produce 50% of the drug's maximal effect. So this is the potency of the drug. This is the concentration or the dose of the drug required to produce an effect to the subject. For question number 5, this is a blank of a drug is the ability of the drug to elicit a response when it binds to the receptor. So this is the efficacy or the intrinsic activity of the drug. It is the ability of the drug to elicit a response when it binds to the receptor. So again, for drug activity, you have 2 requirements. The first requirement is the affinity, and the second requirement is the intrinsic activity or the efficacy. For question number 6, there are 4 types of proteins that are targeted by drugs: the enzymes, the carriers, the ion channels, and the receptors. True or false? So this is true. Now there are 4 types of proteins that are considered to be the targets by the drugs. Question number 7. A drug is said to be blank when it produces a measurable physiological or pharmacological response characteristic of the receptor. Choices: agonist, antagonist, latent, potent. So the correct answer for this is agonist. So an agonist drug is a drug that produces a measurable effect or response that is characteristic of the receptor. So basically, the agonist will mimic the action of the natural ligand to its receptor. For question number 8, the blank of a drug is its ability to bind to a receptor. So the correct answer for this is the affinity. Affinity of the drug is its ability to bind to a receptor, and this is the first requirement for drug activity. For question number 9, this is a blank. A blank is a component, macromolecule of a cell that is located on or inside a cell that interacts with the drug. So the correct answer for this is the receptor. It is the component of the cell. Again, the receptor can be located in the membrane, so that is known as the membrane receptor. It can also be located within the cell, so that is referred to as the intracellular receptor, the nuclear receptor, or the cytosolic receptor, also known as the transcription factor receptor. So all of this are considered to be the receptor. Question number 10. Blank is the measure of the strength of the bond between the drug and its receptor. So the correct answer for this is the affinity. Now it is a measure of the strength of the bond between the drug and the receptor. So of course, this is measured in terms of the Kd, or the measure of the dissociation constant between the drug and the receptor. So it is also related to potency, because the higher the binding, the less would be the amount of the drug that would be required to produce an effect of a specified intensity. Now, the lower the binding, or the lower the strength of the binding, the higher would also be the dose of the drug that would be required to produce the EC50. For question number 11, blank is the ability of the drug to bind to only one receptor. Choices: specificity, selectivity, intrinsic activity. So the correct answer for this is specificity. Question number 12. When the drug interacts with the receptor but does not produce any series of events, thereby blocking the potential action, it is called a blank. So the correct answer for this is antagonism. So the interaction between the drug and the receptor, when the drug would block the potential action of the agonist, is known as the antagonism. And that drug is known as an antagonist drug. And again, when we say antagonist drug, it has affinity to the receptor, but it will not produce an effect. It has no intrinsic activity. So the value of its intrinsic activity is equal to 0. Based on our lecture, there are two types of the antagonist drug, the non-competitive and the competitive. So question number 13, the blank is the concentration of a drug required to produce an effect of a specified intensity. So the correct answer for this is potency. Potency is a property of the drug, and it is usually calculated as the concentration, or the dose, required to produce 50% of the drug's maximal effect. So that is expressed in EC50 or ED50. EC50, so when we say EC50, that is usually used in vitro studies only. So that is usually conducted in the laboratory. So when a drug's potency is measured in a population, animal studies or human populations, another parameter is that we usually use is the ED50, or the median effective dose. So ED50 is the dose that produces the desired effect, typically quantal, and effect in 50% of the population. So when applying these concepts to clinical settings, potency merely indicates the dose of the drug. And of course, when we are going to compare that with efficacy, the efficacy indicates the magnitude of the response, regardless of the dose. For question number 14, these are protein receptors that require the drug to cross the plasma membrane. These are termed collectively as blank receptors. So the correct answer for this is the intracellular receptors. So we can also refer to this as the cytosolic receptors, the nuclear receptors, or the transcription factor receptors. So again, the ligand of these particular receptors are considered to be lipid soluble because they will be able not to cross the plasma membrane. They are considered to be lipid soluble, non-polar, or lipophilic. So example of this are the steroid hormones. Steroid hormones are able to cross the plasma membrane to interact with the intracellular receptors. So again, there are four major types of the receptors. Now, the GPCR, the ligand-gated ion channel, and the enzyme link receptor. Those three are considered to be, it would have a ligand that is hydrophilic, whereas for the intracellular receptors, it would require a ligand that is lipophilic or lipid soluble. Blank is the property of the drug to produce the desired or the biological effect. So the correct answer for this is efficacy. So when we say efficacy, that is the property of the drug to produce a desired effect. It is often used when comparing or evaluating drugs in clinical trials.